Welcome to this lecture on the fundamentals of genetics. Today, we're going to explore a fascinating and somewhat tragic phenomenon known as lethal alleles. We will discover how certain fatal typos in the genetic code can completely alter the fundamental laws of inheritance that Gregor Mendel established. By the end of this session, you will understand why some genetic crosses produce unexpected ratios and how this impacts both human medicine and agriculture. Let us dive in. To begin, let us imagine you are building a complex machine, like a car, using a highly detailed instruction manual. If the manual contains a tiny typo about the color of the seats, the car still runs perfectly fine, just looking a bit different on the inside. However, what if the manual contains a massive, fundamental error regarding how to build the engine? The car will never start, and the entire project fails before it even leaves the factory floor. In the world of genetics, our DNA is that instruction manual. Sometimes a mutation creates an instruction so fundamentally broken that the organism simply cannot survive. These are lethal alleles. Now that we have an intuitive grasp, let us look at the formal definition. A lethal allele is a specific mutant version of a gene that, when present in the required quantity, causes the death of the organism carrying it. Remember that every organism inherits 2 copies of most genes. If an allele is a recessive lethal, the organism must inherit 2 copies, known as the homozygous state, for it to be fatal. If they inherit only 1 copy, they survive, though they might show a different physical trait. There are also dominant lethal alleles, where just a single copy causes death. These are extremely rare because they usually kill the organism before it can reproduce and pass the gene on. You might wonder why we even study lethal alleles if the organisms just die anyway. Understanding lethal alleles is absolutely critical in both human medicine and agriculture. For genetic counselors, knowing about lethal alleles helps predict the likelihood of devastating genetic disorders in families, allowing parents to make informed medical decisions. In agriculture, lethal alleles can cause massive economic losses if breeders unknowingly mate 2 carriers, resulting in dead offspring and smaller litters. Furthermore, the discovery of lethal alleles historically proved to scientists that some genes are absolutely essential for basic survival, not just for determining superficial traits like eye color. Without this concept, we would be completely confused by breeding experiments that produce fewer offspring than expected. This brings us to how lethal alleles alter standard genetic mathematics. In standard Mendelian genetics, crossing 2 heterozygous parents gives a distribution of 4 total parts. But when a homozygous genotype is lethal, those individuals are removed from the surviving population. The denominator of our probability calculations changes from 4 total parts to 3 total surviving parts. The formula shown calculates the ratio of survivors. R represents the ratio of the observable physical traits of the offspring that actually live, and 1 represents the number of heterozygous individuals who carry 1 copy of the lethal allele but survive, which is 2. And 2 represents the homozygous individuals who do not carry the lethal allele at all, which is 1. This shift from a 3-to-1 ratio to a 2-to-1 ratio is the mathematical hallmark of a recessive lethal allele. Let us see this formula in action with the very first discovery of lethal alleles. A scientist named Lucian Quino was breeding yellow mice. He noticed that crossing 2 yellow mice never produced a pure breeding yellow mouse, and litters were unusually small. We're given that the yellow allele, big Y, is dominant for coat color, but recessive for lethality. The normal allele is little y. We set up a cross between two heterozygous yellow mice, big Y little y times big Y little y. The raw genetic combination yields one big Y, two big Y, and one little y. Because the big Y combination is lethal, those embryos die in the womb. We eliminate them from our final count. The final answer shows the surviving offspring will always appear in a ratio of 2 yellow mice to 1 non-yellow mouse. Physically, any living yellow mouse must be a heterozygote. Now that we have seen the example, let us formally prove why this mathematical shift occurs. We are setting out to derive the 2-to-1 ratio from standard Mendelian principles. We start by considering a standard cross between two heterozygous parents, big A times big A. According to Mendel's laws, the expected genotypic distribution is 1 big A, 2 big A, and 1 little a. If we add these up, the total expected offspring parts equal 4. In a normal scenario, where big A is completely dominant and non-lethal, this gives us the classic phenotypic ratio of 3-to-1. This sets our baseline before lethality is introduced. Continuing our derivation, let us now introduce the lethal condition. We assume that the little a genotype is a recessive lethal allele. Because these individuals die before birth, they are completely unobservable in the living population. We must subtract them from our total. The surviving population now consists only of the 1 big A and the 2 big A individuals, giving us exactly 3 total viable parts. When we look at the ratio of surviving phenotypes, we compare the 2 heterozygotes to the 1 surviving homozygote. Therefore, we have mathematically proven that the observable ratio shifts permanently from 3-to-1 down to 2-to-1. This proves why Cugnot saw smaller litters and no pure-breeding yellow mice. To visually reinforce what we just proved, let us look at this Punnett square for the yellow mice experiment. Along the top and left axes, we see the alleles from our 2 heterozygous parents, big Y and little y. Inside the grid, the top left square represents the big Y genotype. Notice the large red X crossing it out? This clearly marks the lethal combination that dies in utero. The 2 yellow squares represent the big Y heterozygotes, which survive and have yellow coats. The single gray square represents the little y homozygote, which survives with a non-yellow coat. This diagram perfectly illustrates how the 4 expected boxes are reduced to 3 viable outcomes, visually confirming our 2-to-1 phenotypic ratio. Now that we understand the mechanics, let us apply this concept to a real-world human condition called achondroplasia. This is the most common form of dwarfism in humans. The allele that causes achondroplasia is dominant for the physical trait of short stature, meaning a person only needs 1 copy to have dwarfism. However, just like the yellow mice, this exact same allele is a recessive lethal allele. If an embryo inherits 2 copies of the dwarfism allele, the skeletal deformities are so severe that the baby will not survive past birth. Because the homozygous dominant state is fatal, it means that all living individuals who have achondroplasia must be heterozygous. Because lethal alleles remove a portion of the expected offspring, we must adjust how we calculate genetic probabilities. This formula calculates the probability of a specific event occurring out of all possible surviving outcomes. P represents the probability of having a child with average height. The numerator, 1, represents the single homozygous recessive genotype that results in average height. The denominator is crucial here. It is 2 plus 1, representing the total number of viable outcomes. We completely ignore the fourth outcome because it is not a surviving possibility. When the variable for viable outcomes decreases, the overall probability of the remaining events increases compared to standard Mendelian genetics. Let us use our probability formula to solve a practical problem. Suppose two people who both have achondroplasia decide to have children. We are asked to find the probability that their living child will have average height. We represent the dwarfism allele as big D and the average height allele as little d. Both parents must be heterozygous, big D, little d. The cross produces one big D, two big D, and one little d. The one big D represents a fatal pregnancy. Since those offspring do not survive, we only look at the three surviving possibilities. The final answer is that there is a 1 in 3 chance, or roughly 33.3%, that their living child will be of average height. This is vital information for genetic counseling. To wrap up our journey, let us review the most important ideas we have covered. First, lethal alleles are mutant genes capable of causing the death of an organism, proving that some genes are absolutely essential for survival. Second, recessive lethal alleles require 2 copies to be fatal, meaning carriers can survive and reproduce. Third, and mathematically most important, the presence of a lethal allele alters the classic Mendelian 3-to-1 ratio into a distinct 2-to-1 ratio among survivors. This shifts our probability denominator from 4 to 3. You have now mastered how to identify and calculate outcomes involving lethal alleles, giving you a powerful tool to accurately predict complex inheritance patterns. Great work!